Assalamu alaikum. Hope all of you are fine. We continue with the embryology lecture. This is a very important lecture, the process of the fertilization. In this lecture, we'll discuss the transport of the gametes, the maturation of the sperms, how the gametes they are viable, the phases or the sequences of the events of the fertilization, then the results or the outcomes of the fertilization, the cleavage of the zygote, and few of the assisted reproductive technologies. Fertilization is a complex sequence of events which takes place when a sperm comes in contact with the secondary oocyte, and it ends when the intermingling of the ends by intermingling of the maternal and the paternal chromosomes. The process of the fertilization lasts about 24 hours, and it takes place in the ampullary part of the uterine tube. Before the fertilization takes place, the gametes they have to be transferred to the ampullary part of the uterine tube. When the oocytes they are released from the ovary, they are captured by the fimbriated end of the uterine tube, and they are carried to the ampullary part of the uterine tube by the peristaltic movement and to the by the rhythmic actions of the cilia lining of the uterine tube. So the your oocytes they will be waiting in the ampullary part for 24 hours for the fertilization to take place. The sperms they have to take a longer course to reach this ampullary part. They have to pass through the cervical canal, the uterus, and then to the uterine tube to reach this ampullary part. So at the time of the ovulation, the cervical mucus it increases in amount and it becomes less thick. So this allows the easier passage of the sperm to be transported through the cervical canal to the uterus and to the oviduct. And also the peristaltic contractions or the muscular contractions of the uterus and the uterine tube, they facilitate the sperm to reach the uterine tube, and also the the tail of the sperm it helps in the movement of the sperm to reach there in the ampullary part. So, if the ovum is present in the ampullary part of the uterine tube, the fertilization will take place. But if there is the ovum has degenerated, then the sperms they will also degenerate in the female genital tract after the. Once the sperms they are present in the female genital tract, they have to undergo a certain conditioning processes which is called the capacitation. And this capacitation is the functional maturation of the sperm, and it takes 5 to 7 hours. So a newly ejected sperm is not able to fertilize the oocyte unless and until it is capacitated, which occurs in the uterus and the oviducts, and it is facilitated by the secretions released by the uterus and the fallopian tubes. So after the capacitation, the sperm is able to undergo the acrosomal reaction. So during the capacitation, which is a functional maturation of the sperm, there is the removal of the glycoprotein coat over the acrosome or the acrosomal cap, and the certain seminal proteins which are present over the acrosome region, they are also removed. So the membrane component of the acrosome membrane that is extensively altered, and so that this membrane is capable of undergoing the acrosome reaction or the release of the enzymes through these acrosomal cap. There is also alterations in the tail of the sperm. That is, it becomes there are changes in its motility, and there are whipping movements of the tail together with the sideways movement of the head, and this, it is referred to as the hyperactivity of the sperm or the hyperactivity of the tail. So these are the changes which takes place during the process of the capacitation, and this capacitation process it enables the sperm to fertilize the egg. So a human oocyte, they are fertilized within the 12 hours of the after the ovulation, and they cannot be fertilized after the 24 hours after which they degenerates. And the human sperms, they also do not survive for more than 48 hours in the female genital tracts. But however, the sperms and the oocytes, if they are taken from the male and the female tracts, they are stored for frozen and stored for many years, and they can be used for the in vitro fertilization. Now, the sequences of the events or the phases or the steps of the fertilization are as follows: that first the sperm has to pass through these corona radiata cells. Then, secondly, it has to cross the barrier of the zona pellucida layer. Then it reaches the plasma membrane of the oocyte, and it comes in contact with the plasma membrane, and then the cytoplasm and the genetic material of the sperm is transferred inside the cell membrane. So first it has to cross this barrier of these corona radiata cells which are surrounding the oocyte. Now, when a capacitated sperm comes in contact with the corona radiata cells, then there is a release of the enzymes from the acrosomal cap, these acrosomal enzymes, and it causes the lysis and of the corona radiata cells, and these corona radiata cells, they are dispersed and make a passage for the sperm to reach the zona pellucida. The enzymes mainly they are the hyaluronidase enzyme which causes the lysis and the dispersal of these corona radiata cells, plus the hyperactivity of the sperm, that is the sperm tail is hyperactive and motile, and the side to side movements of the head of the sperm, they also facilitate its passage through these corona radiata cells. So once it reaches the zona pellucida layer, it comes in contact with the zona pellucida layer. The zona pellucida layer is a thick glycoprotein layer which is made up of the zona proteins, and these ZP3 proteins, they comes in contact with the proteins of the acrosomal head. Then there is also the release of the enzyme from the acrosome, particularly the acrosin enzyme. This acrosin enzyme, it causes the lysis of the zona pellucida layer, and the sperm, it penetrates and reaches the plasma membrane and comes in contact with the plasma membrane. So once it comes in contact with the plasma membrane, the zona reaction takes place. So this is a diagram showing you the acrosomal reaction. You can see here that this is the acrosomal cap, which is made of the inner acrosomal membrane, and it has the acrosomal enzymes in it. So after the changes in the acrosomal region, and there is alterations in the acrosomal membrane, it becomes porous, and it causes the release of the acrosomal enzymes, which are the hyaluronidase and the acrosines, and then this cap is the acrosomal cap is removed after the acrosome reaction takes place. So this is a diagram showing you that the sperm has reached the plasma membrane after crossing the corona radiata cells and the zona pellucida lysis, and it reaches the plasma membrane of the oocyte. So once it reaches the plasma membrane of the oocyte, the cortical enzymes or the lysosomal enzymes, they are released from the plasma membrane, and the lysosomal enzymes which are released from the cortical granules near the plasma membrane, they are released into the perivitelline space. So once these enzymes they are released into the perivitelline space, they changes the permeability of the zona pellucida layer, and the zona pellucida layer becomes impermeable to the millions of the sperms which are surrounding it. So only one sperm is allowed to enter into the plasma membrane. So first the sperm which reaches the plasma membrane, it causes the zona reaction, and the zona pellucida layer becomes impermeable to the rest of the sperms which are degenerated around after the 48 hours embedded in the corona radiata cells. So this is called the zona reaction, that is the impermeability of the zona pellucida layer to the rest of the sperm, and it is facilitated by the enzymes which are released from these cortical granules into the perivitelline space near the plasma membrane. So you can see that here the other enzymes which are released when it passes through the corona radiata, the hyaluronidase enzyme, and also the enzymes, they are also released from the mucosa of the tube, and they also facilitate its passage through the corona radiata, plus the, as we have discussed, the flagella action of this sperm. It also helps its penetration. And there are various enzymes which are released from the zona pellucida when it comes in contact with the zona pellucida from the acrosome, but the esterases, the acrosine, and neuraminidase, which causes the lysis of the zona pellucida. But the most important one is the acrosine. So this is the diagram showing you clear picture. So the next is the fusion of the cell membrane of the ovocyte and the sperms. So the head and the tail of the sperms, they enter into the cytoplasm of the oocytes, but the plasma membrane and the mitochondria, which are in the middle piece of the tail, they remain outside. So the genetic material is injected into the inside, and it forms a male pronucleus, and a female pronucleus is formed. And after that, there is the fusion of the genetic material, or the completion of the second meiotic division, and the female pronucleus is formed following the decondensation of the maternal chromosomes, and this second meiotic division is completed, and the initiation of the cleavage takes place. Now, this zygote is which is formed, which is a pluripotent cell, and or a totipotent cell, and it undergoes various cell divisions, and along with the cell divisions, it also start moving towards the uterine cavity. So this is all the events showing you the formation of the spindles, and this zona pellucida is still here, and these female pronucleus formation, then the male pronucleus, and the revival of the second meiotic division, and then is the initiation of the cleavage. So after the fertilization, there are certain facts about the fertilization, that it is completed within 24 hours of the after ovulation, and after the 24 hours, the female the oocyte, it cannot be fertilized, it degenerates. So approximately 400 to 600 million sperms, they are deposited at the cervical opening during ejaculation, and some sperms, they are present in the folds of the cervix, and they are gradually released into the cervical canal, and this gradual release of the sperms from it increases the chances of the fertilization. So after the 48 hours, they are not able to survive in the female genital tract, so they degenerate. So only about 200 sperms, they reach the fertilization site, and most, they degenerate and absorbed by the female genital tract. So as a result of the fertilization, the secondary oocyte, it completes its meiosis, that is the second meiotic division is completed. The normal or the diploid number of the chromosomes, that is the 46th number of the chromosomes, is restored, and it enables the process of the genetic variability, or the variation of the human species, as the maternal and the paternal chromosomes, they intermingle with each other. So there is a chance of the genetic variations. So there is also the sex of the embryo is determined at the time of the fertilization. So the embryo, it contains only the maternal mitochondria, because the sperm mitochondria is dispersed into the egg cytoplasm and discarded. So the zygote, it contains only the maternal mitochondria. Now, the process of the cleavage. So, you can see in the diagram, this is the ampullary part of the fallopian tube where the fertilization takes place. And as a result of the fertilization, the zygote is formed, which is a totipotent cell. So, this zygote, it undergoes the repeated mitotic divisions, which are called the cleavage. And so it forms first a 2-cell stage, the 4-cell stage, the 8-cell stage, and then a ball of the cells, which is called the medulla, and then a fluid-filled cavity which is called the blastula. So the cleavage of the human zygote, it occurs all in the uterine tube or the fallopian tube, and it is holoblastic, that is, it divides the zygote completely into the daughter cells or the blastomeres. You can see here also in detail that the first cleavage, it takes place about 30 hours after the fertilization, and it divides longitudinally into the two blastomeres. Now each blastomere is a pluripotent cell, and they are capable of forming the embryo. So the one is slightly larger than the other. The second cleavage takes place at the right angle to the first one, and it forms the four blastomeres. The third is take place at the 72 hours, then the 8-cell stage, unless and until it forms the medulla, which is a 16 to 32 cell stage. And then a fluid-filled cavity is formed, which is called the blastocyst or a blastula. Now you can see here that is at the end of the fourth day, the marula embryo reaches the uterus, and it looks like a mulberry, and it is known as the marula, and it is a 16 to 32 blastomere cells, and these internally they are surrounded by the trophoblastic cells, and it is formed 3 days after the fertilization, and as it is entering towards the uterus. So what are the significances of the cleavage? So the cleavage process converts a unicellular zygote into a multicellular embryo. It maintains the cell size and the nucleocytoplasmic ratio of the species. The cleavage produces the large member of the cells or the blastomeres, which are required for the building of the offspring's body. During the cleavage, the quick mitotic divisions of the blastomeres occurs, following which there is no growth of the blastomeres. The cleavage, it brings about this distribution of the cytoplasm among the blastomeres. Now the types of the assisted reproductive technologies or the ART. There are various assisted reproductive technologies like the cryopreservation of the embryos, intracytoplasmic sperm injection, assisted in vivo fertilization, and the surrogate mothers. Now, what is this assisted reproductive technology? It is a technology which is used to treat the infertility, and it includes the fertility treatments that handle both a woman's egg and a man's sperm. So it works by removing the eggs from a woman's body. That is, after the hormonal stimulation of the ovaries, the mature oocytes, they are released at their ovulation, and they are taken up. And then these eggs, mature eggs, they are placed in a petri dish where they are fertilized with a capacitated sperm, and after the fertilization in vitro, this process of the cleavage is initiated in this culture medium, and the forms the 2-cell stage and 4-cell stage and 8-cell stage. So at the 8-cell stage, these each blastomere, it is capable of forming the embryo. So they are taken up from these petri dish, and they are transferred from these cleaving embryos into the uterine cavity through a catheter which is inserted through the cervix and cervical canal into the uterine cavity. So there are more number of these blastomeres or these cleaved cleaving these zygotes. They have been transferred into this area or the uterine cavity. So this in vitro fertilization is this most common and the effective type of the assisted reproductive technology. So some procedures of this ART, they use the donor eggs, the donor sperms, or the previously frozen embryos also. It may also involve a surrogate or a gestational carrier. A surrogate is a woman who becomes pregnant with a sperm from a male partner of the couple. And a gestational carrier becomes pregnant with an egg from a female partner and the sperm from the male partner. So the most common complication of this ART is the multiple pregnancy because not a single this cleaved blastomeres they are used and they are planted in the uterine cavity. So to for the success rate, more than one is placed in the uterine cavity. So this can be prevented or minimized by limiting the number of the embryos that are put into the woman's body. So this is a diagram showing you the process of cleavage in the in vitro fertilization. You can see that this is the two cell stage, and there are a lot of these sperms which are degenerating in outside and attached to these corona radiata cells. You can visualize here. And these are degenerating sperms. The polar body, which is non-functional, it is only required for the nutrition of the developing oocyte. So this is the all about the fertilization and the assisted reproductive technology and the process of the cleavage. So if you have any difficulty in this lecture, you can ask me on the WhatsApp group. So thank you. It's all about the fertilization. Next, we'll continue with the blastula or the blastocyst formation.